You have probably noticed it. A cut that would have healed in 3 days when you were 20 now takes a week. A bruise that would have faded in days lingers for 2 weeks. The skin that once snapped back after a scrape now seems to deliberate before closing. Most people assume this is simply because the body slows down with age, that cells become less energetic, less motivated, less capable, that aging is a kind of generalized deceleration. That assumption is wrong, and the real explanation is stranger, more specific, and more consequential than a simple slowdown. The real reason your wounds heal slower as you age is not that your cells become weaker. It is that your tissue is filling up with cells that refuse to die, cells that have stopped functioning but cannot be eliminated. Cells that sit in your tissue like biological squatters, secreting a toxic cocktail of inflammatory signals that actively poison the repair process happening around them. Scientists call these cells senescent cells. Everyone else, once they understand what they do, calls them zombie cells. And they are one of the most important and most underappreciated drivers of biological aging. To understand what a senescent cell is, you need to start with a fundamental constraint of cellular biology. Most cells in your body can only divide a limited number of times. Each time a cell divides, copying its DNA and splitting into 2 daughter cells, the very tips of its chromosomes, structures called telomeres, get slightly shorter. Telomeres are protective caps of repetitive DNA sequences that sit at the ends of chromosomes, preventing the chromosome ends from being recognized as damaged DNA and from fusing with other chromosomes. They function like the plastic tips on the ends of shoelaces, protecting the functional genetic information from fraying with each replication. And with every cell division, a small portion of the telomere is lost because the DNA replication machinery cannot fully copy the very end of a linear chromosome. This progressive telomere shortening was discovered and characterized by Leonard Hayflick in the 1960s, and the observation that normal human cells can only divide approximately 50 to 70 times before stopping is called the Hayflick limit. When a cell's telomeres shorten to a critical threshold, the cell detects this as a damage signal. Critically short telomeres look to the cellular surveillance machinery like a broken chromosome end, like DNA damage requiring repair. And when the cell detects this apparent damage, it activates a set of tumor suppressor pathways, primarily involving proteins called p53 and p21 that arrest the cell cycle. The cell stops dividing. Permanently, it enters a state called cellular senescence. And here is the first twist that makes senescence so biologically interesting. The cell does not die. It stops dividing, but it stays alive. It remains metabolically active, consuming nutrients, producing energy, continuing to synthesize proteins, but it no longer replicates. It no longer contributes to tissue renewal. It just sits there. And what it does while it sits there is what makes it so destructive. Senescent cells develop a characteristic behavior pattern called the senescence-associated secretory phenotype, or SASP. The SASP is a dramatic shift in what the cell produces and secretes into its surrounding tissue. Senescent cells begin pumping out large quantities of pro-inflammatory cytokines, including interleukin-6, interleukin-8, and tumor necrosis factor alpha. They secrete matrix metalloproteinases, enzymes that break down the structural proteins of the extracellular matrix, the collagen and fibronectin scaffolding that tissues depend on for their physical integrity. They secrete growth factors that can paradoxically promote abnormal cell proliferation in neighboring cells. And they secrete signals that can induce senescence in neighboring healthy cells, spreading the senescent state through the tissue in a process sometimes called paracrine senescence, or the bystander effect. Think about what this means for wound healing. When you sustain a wound, the repair process depends on a precisely coordinated sequence of cellular events. Platelets arrive first and form a clot. Immune cells clear debris and pathogens. Fibroblasts migrate into the wound site and begin synthesizing new collagen to rebuild the extracellular matrix. Epithelial cells proliferate and migrate across the wound surface to close the gap. Stem cells in the surrounding tissue are activated to produce new cells, replacing those lost to injury. This is an elegant, tightly regulated biological program, and it depends critically on the cells involved being responsive to the signals that direct each step. Now, place this repair program in a tissue saturated with senescent cells, secreting SASP. the inflammatory cytokines from senescent cells chronically activate the immune cells that should be performing a brief, targeted response and then standing down. Instead of a clean inflammatory phase, followed by resolution and repair, the tissue remains in a state of chronic, low-grade inflammation. The matrix metalloproteinases, secreted by senescent cells, degrade the collagen being laid down by fibroblasts, undermining the structural scaffold that the wound needs to close. The SASP signals interfere with the proliferative capacity of the fibroblasts themselves, making them less responsive to the growth signals that should drive their migration and collagen production. And the paracrine senescence effect means that some of the cells recruited to the wound site may themselves be pushed into senescence by the CSP environment, reducing the pool of functional repair cells available. The wound heals, but it heals slowly, incompletely, and with more scar tissue and less functional tissue restoration than in a young, senescence-free environment. This is the mechanism behind the slower wound healing of aging, not a simple deceleration, a specific molecular interference, zombie cells poisoning the repair process with their secretions. But why does the body accumulate senescent cells with age in the first place? The answer is a story about a trade-off, and it is one of the most revealing trade-offs in all of evolutionary biology. Cellular senescence, the very process that causes all this damage in aging tissue, exists in the first place as a tumor suppression mechanism when a cell sustains DNA damage, whether from telomere shortening, from oxidative stress, from radiation, or from oncogene activation. The same p53 and p21 pathways that drive senescence are activated to prevent that potentially cancerous cell from dividing and propagating its damage. Senescence is the cell saying, "I am too damaged to divide safely. I will stop here. I will not contribute to a tumor." In the short term, in a young body, this is enormously protective. Cells that might otherwise become the founding cells of a cancer are arrested and removed from the replicating pool. The senescence response is part of why cancer rates are relatively low in young people, despite constant DNA damage occurring in trillions of dividing cells. The problem is clearance. In a young, healthy immune system, senescent cells are recognized and eliminated by immune cells, particularly natural killer cells and macrophages, which detect specific markers on the surface of senescent cells and destroy them before their SASP can accumulate to harmful levels. The immune system performs ongoing surveillance and clearance of senescent cells, keeping their numbers low in healthy young tissue. But as the immune system itself ages, this senescent cell clearance becomes less efficient. Fewer natural killer cells, less responsive macrophages, declining immune surveillance capacity. And as clearance declines, senescent cells accumulate. They build up in tissues throughout the body. Their collective SASP output rises. The chronic low-grade inflammation they generate, a state researchers have named inflammaging, drives the tissue dysfunction associated with biological aging. This is the evolutionary trade-off at the heart of aging biology. The senescence response was selected by evolution because it protects young organisms from cancer during their reproductive years. The accumulation of senescent cells in later life, when immune clearance is compromised, was not a strong target of natural selection because most organisms in evolutionary history did not survive long enough for this accumulation to become the primary driver of their deterioration. We live long enough now for the trade-off to become visible, long enough to see the anti-cancer mechanism of our youth become the driver of our aging, long enough to feel it every time a wound takes twice as long to close as it did 20 years ago. The SASP does not only affect wound healing, it affects every tissue that accumulates senescent cells. In the joints, senescent cells in the cartilage and synovium secrete matrix metalloproteinases that break down articular cartilage, contributing to the development of osteoarthritis. In the cardiovascular system, senescent endothelial cells and smooth muscle cells secrete inflammatory cytokines that promote arterial stiffening, plaque instability, and atherosclerotic progression. In the brain, senescent microglia and astrocytes contribute to neuroinflammation that impairs neuronal function and is increasingly implicated in the pathophysiology of Alzheimer's disease and other neurodegenerative conditions. In the lung, in the liver, in the kidney, in the skin, in the muscle, the pattern is the same. Accumulating senescent cells, rising SASP, chronic tissue inflammation, declining function. The zombie cells spread their influence across the entire aging organism. And here is where the story opens into one of the most exciting frontiers in biology today. Because if senescent cell accumulation is a primary driver of aging and age-related disease, then eliminating senescent cells might reverse or slow some of the biology of aging. This is the premise behind a field called senolytic therapy. Senolytics are drugs or compounds that selectively kill senescent cells while leaving normal cells unharmed. They exploit the specific vulnerabilities that senescent cells develop in their arrested state. Senescent cells, unable to divide and unable to die normally through apoptosis, often develop an enhanced dependence on specific anti-apoptotic survival pathways that protect them from the programmed cell death signals that would otherwise eliminate them. Senolytic drugs target these survival pathways, disabling the protection that allows senescent cells to persist and triggering their death. The results in animal studies have been remarkable. In mice, periodic clearance of senescent cells using genetic tools or senolytic drugs has extended healthy lifespan, delayed the onset of multiple age-related diseases, improved physical function, restored aspects of organ function, and in some studies, rejuvenated tissue repair capacity, including wound healing speed. A single treatment course of senolytics in aged mice has been shown to improve cardiovascular function, lung compliance, physical endurance, and markers of frailty. The effects are not merely symptomatic. They appear to reflect genuine improvement in the biological age of the treated tissue. Human clinical trials of senolytic compounds are now underway, and early results have been published for several conditions. Dasatinib and quercetin, one of the most studied senolytic combinations, have shown evidence of senescent cell reduction in human adipose tissue and lung tissue, with associated improvements in physical function measures in patients with idiopathic pulmonary fibrosis, a devastating age-related lung disease previously considered irreversible. The field is young, the evidence is early, and significant questions remain about dosing, safety, tissue specificity, and long-term effects. But the basic biology is compelling enough that it has attracted serious scientific and investment attention as a potential approach to treating not individual diseases of aging, but the underlying biology that drives them all. The wound that heals slowly is not just an inconvenience. It is a readout, a visible, tangible signal of a cellular process unfolding in your tissue, accumulating quietly for decades, reshaping the biology of your organs and your immune system in ways that will determine your health trajectory over the remaining decades of your life. The zombie cells in your skin are the same zombie cells accumulating in your joints, your arteries, your brain, your lungs. The slowness of your healing is the surface manifestation of something running much deeper, something that science is only now beginning to understand well enough to target. And the deepest insight from all of this is what it reveals about the nature of aging itself. Aging is not entropy. It is not the body simply wearing out like a machine, accumulating damage with use. Aging is biology. It is the product of specific molecular mechanisms, specific cellular programs, specific evolutionary trade-offs that made sense for organisms that lived shorter lives in different environments. The senescence program is not a flaw in your design. It was a solution to a problem, cancer prevention, that worked well for millions of years. The accumulation of its side effects in long-lived organisms is a consequence of a world that evolution did not fully anticipate. We live longer than the design specification assumed. And some of the most important work in biology right now is figuring out how to update the design, how to clear the zombie cells, how to restore the clearance mechanisms that kept them in check when we were young, how to give the tissue repair program the clean environment it needs to run at its original efficiency. That is the biology behind the wound that takes too long to close. Senescent cells, SASP, the evolutionary trade-off between cancer suppression and tissue maintenance, and the emerging possibility that this trade-off, written into our biology over millions of years, might finally be something we can intervene in. That is applied biology, running in every healing cut, every slow-closing bruise, every stiff morning joint, telling you the story of your own cellular aging in the language of the body you live in. There is one more dimension of this story that deserves attention, because it connects the biology of senescent cells to a question that most people have never thought to ask. Why do senescent cells develop the SASP in the first place? Why does a cell that has stopped dividing begin secreting inflammatory signals? It seems counterproductive. A broken cell making itself more visible and more destructive seems like a design failure. But there is an evolutionary logic to it. and understanding that logic changes the way you think about what the SASP actually is. The SASP is, at its origin, a signaling system. When a cell enters senescence due to DNA damage, the inflammatory signals it secretes serve to recruit immune cells to the site. The cytokines say, in molecular terms, something has gone wrong here, send help. In a young organism with a functional immune system, this is useful. The immune cells arrive, recognize the senescent cell through its surface markers, and eliminate it. The SASP is the distress signal. The immune clearance is the response. The system works as designed when both components are functional. The senescent cell calls for its own elimination. The immune system answers. The cell is cleared. The tissue remains healthy. The problem emerges when the immune response fails to answer the call, when the natural killer cells that should be eliminating senescent cells are fewer in number, less active, less capable of recognizing senescent cell surface markers. The senescent cell continues to secrete its SASP, calling for help that does not arrive. Its signals accumulate in the tissue. Neighboring cells are exposed to chronically elevated inflammatory cytokines. Some of them enter paracrine senescence. They too begin secreting SASP. The signal amplifies without resolution. What was designed as a brief distress call and response becomes a permanent alarm, ringing in tissue throughout the aging body, attracting chronic immune activation, degrading the extracellular matrix, disrupting the repair programs that depend on a clean tissue environment. This reframing changes the therapeutic target. The goal is not simply to kill senescent cells, though that is one valid approach. The goal might also be to restore the immune clearance capacity that was supposed to eliminate them in the first place. Aging of the immune system, what immunologists call immunosenescence, is itself driven partly by the accumulation of senescent immune cells. Immune cells that have undergone senescence themselves, that have reached their own Hayflick limit, or been driven into senescence by the chronic inflammatory environment created by SASP, are less capable of clearing senescent non-immune cells. It is a feedback loop. Senescent cells impair immune clearance. Impaired immune clearance allows more senescent cells to accumulate. More senescent cells further impair immune function. The system spirals. The aging process accelerates its own pace. Exercise has direct and measurable effects on this spiral. Regular aerobic and resistance exercise reduces the burden of senescent cells in multiple tissues. It does so through several mechanisms. Exercise acutely increases the activity of natural killer cells, the primary immune executors of senescent cell clearance. Exercise reduces chronic inflammation, lowering the background SASP environment that drives paracrine senescence. Exercise stimulates the production of anti-inflammatory myokines, signaling molecules released by contracting muscle that have systemic anti-inflammatory effects. And exercise, by maintaining metabolic health and reducing visceral adiposity, reduces the chronic oxidative stress that is one of the primary drivers of DNA damage and premature cellular senescence. Visceral fat, the metabolically active fat depot surrounding the abdominal organs, is particularly rich in senescent cells and in SASP-secreting cells. Adipose tissue senescence is one of the most important sources of the systemic chronic inflammation that drives age-related disease. Reducing visceral fat through exercise and dietary change directly reduces the senescent cell burden in one of the most influential tissue depots in the body. The anti-aging effects of exercise, which are among the most consistent and largest magnitude effects observed in longevity research, operate partly through exactly this mechanism, reducing senescent cell accumulation and restoring immune clearance capacity. Caloric restriction and its molecular mimetics, compounds like rapamycin that activate the AMPK and inhibit the mTOR signaling pathways associated with caloric restriction response, also show evidence of reducing senescent cell burden and SASP in animal models. The longevity effects of caloric restriction, some of the most robust and reproducible effects in aging biology, appear to operate at least partly through reduced cellular senescence. Whether the same mechanisms are accessible through dietary interventions in humans without the practical constraints of severe caloric restriction is an active area of research, and one with significant implications for translating the animal biology into actionable human health guidance. The final thought worth sitting with is this. Every cell in your body is engaged in a continuous negotiation between its own survival and the survival of the organism it inhabits. Cellular senescence is one expression of that negotiation. A cell that detects it is too damaged to divide safely, chooses to stop rather than risk producing a cancerous daughter cell. It sacrifices its own reproductive future for the protection of the whole. This is a form of biological altruism, encoded in the tumor suppressor pathways of every cell in your body. The tragedy of aging is that this altruistic act has a cost. The cells that stop dividing to protect you from cancer do not simply disappear, they linger. Their SASP accumulates. Their presence degrades the tissue around them. And over decades, the accumulation of their sacrifice becomes one of the primary forces eroding the function of the body they were trying to protect. That is the biology of zombie cells, not horror fiction, not metaphor. Cells doing exactly what they were designed to do, and the body paying the price of that design in the currency of slower healing, stiffer joints, declining organ function, and the accumulated weight of decades of cellular senescence. Applied biology, running in every tissue, in every aging body, written in the language of cells that refuse to die so that you could live. That is the real reason your wounds heal slower. And now you know exactly why. Let me take you one step further into the specific cellular machinery that makes senescent cells so difficult for the aging body to eliminate. Because understanding why they persist is as important as understanding what they do. And the answer involves one of the most fundamental cellular survival mechanisms in biology. Normal cells, when they receive signals indicating they are damaged beyond repair, or when they are no longer needed by the tissue, undergo a process called apoptosis. Apoptosis is programmed cell death, a highly organized, energy-dependent process in which the cell systematically dismantles itself, packages its contents into membrane-bound fragments, and signals to neighboring immune cells to clear the debris. Apoptosis is clean, controlled, and essential for tissue homeostasis. It is how the body removes cells that are no longer functional without triggering inflammation. And it is the process that senescent cells have specifically learned to resist. Senescent cells upregulate a family of proteins called BCL2, anti-apoptotic proteins. BCL2 and its family members, including BCLXL and BCLW, normally function as survival signals, binding to and inhibiting the pro-apoptotic proteins that would otherwise trigger cell death. In normal tissue, the balance between pro-apoptotic and anti-apoptotic signals determines whether a cell lives or dies. In senescent cells, this balance is tipped strongly toward survival by the chronic upregulation of BCL2 family proteins. The cells become, in a molecular sense, resistant to death. They suppress the very apoptotic machinery that should be eliminating them. And this resistance is what allows them to persist in tissue for years, secreting SASP, accumulating in growing numbers, contributing to the inflammatory environment of aging tissue. This is precisely the vulnerability that senolytic drugs exploit. The first generation of senolytics, including the combination of dasatinib and quercetin and the compound navitoclax, work primarily by inhibiting BCL2 family proteins. By blocking the anti-apoptotic shield that senescent cells depend on for survival, these drugs restore the sensitivity of senescent cells to apoptotic signals, allowing them to die through the same programmed death pathway that other damaged cells use. Because normal, healthy cells maintain a more balanced ratio of pro- and anti-apoptotic signals and do not depend as heavily on BCL2 family overexpression for survival, they are less affected by the senolytic treatment. The selectivity, though imperfect, is real enough to produce measurable reduction in senescent cell burden without proportional damage to healthy tissue. The next generation of senolytics is attempting to improve this selectivity by targeting other specific vulnerabilities of senescent cells, including their altered metabolism, their characteristic surface markers, and the specific signaling pathways that maintain their arrested but metabolically active state. Chimeric antigen receptor T cells, the same cellular engineering technology being developed for cancer therapy, are being explored as a potential tool to direct immune cells to recognize and eliminate senescent cells with high specificity. The field is moving rapidly, driven by the recognition that senescent cell accumulation is not a peripheral feature of aging biology, but a central mechanism driving the functional decline across multiple organ systems simultaneously. What this means for the future of medicine is significant. Most of the major diseases of aging, cardiovascular disease, neurodegeneration, osteoarthritis, metabolic dysfunction, pulmonary fibrosis, are currently treated as separate conditions with separate drugs and separate clinical approaches. Senolytic therapy offers the possibility of a different kind of intervention, one that targets the shared underlying biology across all of these conditions simultaneously. Remove the senescent cells, reduce the CASP, lower the chronic inflammatory burden, restore the tissue environments in which repair programs and stem cells can function at something closer to their youthful capacity, and you may shift the trajectory of multiple age-related conditions at once, not by treating the symptoms of aging, but by addressing one of its molecular drivers. That is the promise. The reality is that biology is complex, that aging is multifactorial, that no single intervention will reverse all of its dimensions, and that the translation from remarkable animal results to meaningful human benefit is a process that has humbled many promising research directions before. The zombie cell hypothesis of aging is well supported by evidence, but the degree to which senescent cell clearance alone can extend healthy human lifespan or meaningfully reverse established age-related disease in humans is still being determined. What is not in doubt is the mechanism. Senescent cells accumulate. Their SASP degrades tissue function. Their persistence is maintained by specific molecular survival pathways, and their accumulation is measurably connected to the functional decline of aging tissue, including the tissue that is supposed to close your wounds. The cut on your hand that is healing slowly right now is a biological message. It is the readout of decades of cellular decision-making, of telomeres shortening, of DNA damage accumulating, of cells entering senescence to protect you from cancer, of immune clearance declining, of CASP signals rising, of fibroblasts working in a contaminated environment. Every day that cut takes to close is the phenomenology of all of that molecular history, made visible on the surface of your skin. Now you know what is happening underneath, and knowing the mechanism is the first step toward the possibility of changing it. Applied biology in the slowness of your own healing. The zombie cells, finally explained,